Hi, I'm Eric Chudler. I'm a neuroscientist at the University of Washington. And today I'm speaking with Dr. Margie Byers, who is in the Department of Anesthesiology and Pain Medicine at the University of Washington. Hi, Margie.
Hi, Eric.
I was thinking that we have known each other for over 40 years. When I
Yeah,
started at the University of Washington as a graduate student in 1980, you were already on faculty.
right.
So you were here before that. So tell me a little bit about your pathway to Seattle and how you
Okay.
got to where you are now.
Well, I got to thinking about this when you sent some sort of general questions. And a lot has depended on some very good accidents that I fell into neuroscience. In college, my goal was to figure out what I was going to do after college, and I was still adrift as a senior in 1962. And by chance, I took a course with Keith Porter on cell biology and electron microscopy that included a lot about nerve cell biology as well. And I just fell in love with it instantly. And within 3 days, I had looked through all their histology slides, looking around for something else to do. And I got put onto a group project studying how nerve cells regenerate. And so this is almost 60 years ago, if I've got the numbers right. And we didn't know a heck of a lot. I mean, messenger RNA had not yet been found. And by the time I took those courses and got into graduate school, I got into axonal transport. It was a whole new way to study nerve cells. And so when eventually we got to Seattle in 1971, my husband Breck had a job. I didn't, but I started walking around going to seminars and found both Anita Hendrickson and Ray Lund doing wonderful work, and went and talked to them. And one thing led to another, and I joined Ray Fink's research group using axonal transport to see whether local anesthetics affected, and indeed they did. So if the anesthetic was too high a concentration, this essential way by which a nerve cell keeps itself alive was slowing down or stopping. And so in that era at the UW, NIH would give a huge grant to a place like the Anesthesia Research Center or the Dental School Research Center, and then they could give pilot projects to new people. It was a great way to start a career. And I was only working 3 partial days a week at the beginning with little kids at home, but by 1979, I was here full time. I had NIH funding, but it took a while to grow a career. And then I went on from project to project over the years. And again, accidents really were important. We were smart enough to pay attention. Why was something not working? And one of these was Chuck Kimberly, a wonderful student from the dental school, who made some cavities in rat teeth and wanted to see how everything changed where those nerve fibers come into the brainstem. And he couldn't make heads or tails out of it. One thing was up, one thing was down, and it made no sense. So he went to look at the teeth. He thought, well, maybe I didn't do my rat dentistry very well. And lo and behold, here the nerve fibers were sprouting all over the place. And that just opened up a couple decades of research. What nerve fibers react and why are they reacting? Turns out they're very important to get inflammation working properly, to promote wound healing. The rat molars would be quite well healed within a few weeks, but not if the nerves have been disconnected. So starting about 1975, I started going to neuroscience meetings, and they were absolutely wonderful. I felt like it was a learning time for me. I'd have a poster or a talk, but you could just see what was going on in the field. And I did that for 30 years. And after 30 years, I was drifting into a situation where instead of going to the meeting, I'd go and visit college friends who lived in that town. And I'd go to art museums. I thought, now, wait a minute, maybe I should be back in Seattle, and I was due to retire at the end of 2006. So I am in my 15th year of retirement, still coming here. I had great students all the way through and including through 2010. And then worked with some wonderful colleagues here on papers, and actually have just had another one accepted. So I brought some show and tell. I don't know if this will work, so you can tell me if it doesn't work.
Okay, we can have a look at it, yeah.
Yeah, but the situation is that the funding led us into the trigeminal nervous system, which is everything in the face, sensory innervation mostly, but some motor. I have been in the peripheral nervous system for my whole career. And that involves also studying tissues and how are the tissues and the nerves talking to each other, what do they do? So I have a poster about the trigeminal nervous system. Let's see if this works.
Yeah, I can see it.
Yeah, so look at all the different tissues that are getting sensory input from this one big sensory ganglion, the fifth cranial nerve. So that's kind of an overview, but then this is a diagram of the innervation of teeth from 20 years ago. And I've colorized the dentin as yellow in the tooth. The tooth is somewhere here. You have pulp, dentin, enamel on top, and all these tissues down below. And I'm actually going to be in a meeting with people in the dental school in a couple of days who want to learn about all those tissues around the root. Because when they try to move teeth for orthodontia, take off the braces, the teeth go right back to where they were. And they're still trying to solve that. We have a new paper in which we've identified a half a dozen, don't know where the figure is, a half a dozen different kinds of innervation just of the dentin. But a couple of years ago, we published this picture where the green ending is like a complex touch receptor, and it's it's on the edges of teeth. So we'd always been kind of going right through the middle of the tooth, and that should be good enough. And no, once we started doing a whole series through, we could find other nerve fibers. So the other thing I do, and you can see the massive paper all over the place, is try to write papers. So this one made it into the lap of Elsevier Publishers just yesterday. And this is what it looks like. So they got it in February. They had a lot of complaints. And now when I send it back, I turn it yellow wherever we've tried to fix something. So we changed the figures quite a bit. This was another part where they didn't like what we had said at all. So it's all yellow, and get to the discussion. The discussion was better. So, and actually I need to thank you, Eric, for showing me a brand new paper that is just fabulous on what neuroscience can do nowadays. And this is a group that on March 26 published that they had identified a molecule that was essential for tooth pain if the pain was triggered by cold. And so this is a brand new era. So the last 5 years or so, I've really gotten more serious about cleaning up. And of course, I keep finding something else that we had forgotten about and never wrote up. But I'm just having a wonderful time seeing what all the new techniques can do, the optogenetics and these amazing ways to study nerve cells. In a way, I wish I could go to a neuroscience meeting, except I'm not a great traveler anymore.
Yeah, well, so, I know that whenever I read one of your papers, I always think that they're literally a work of art because of the photographs and figures in there. They're just so beautiful to look at. So your papers are just so beautiful in an artistic sense.
Well, that's what got me into this, because I'm much more artistic than scientific. So you have to sort of drag me to the microscope to count things, but I will do it. But mostly I'm just having a ball, having a way to be scientific in a visual way. Yeah.
And,
So
and is art something you do in your spare time, or I know
it
you're in
is.
the lab most of the time, but not in the lab,
I'm,
what do
I'm
you
a,
do?
well, I mean, I do these diagrams for one thing, and really have fun with it. Um, but I love to do watercolors, and music is another big thing. And we were just talking with our family in Portland, Oregon, about how great it has been over the years to jump in the car and go to Mini Theater. It's like a 6-minute drive. The logistics are incredibly easy compared to my sister who lives in New York, and it takes them 2 hours to get to the Met Opera. So we've been to many, many, many concerts at Mini, and hiking, lots of hiking in the past 14 months with the pandemic, simply within our neighborhood. And occasionally we go see a friend who lives 3 miles north, and it seems like a giant expedition to not be in our little neighborhood. So I just feel very lucky to have been able to keep doing this. And the UW has been open. This corridor has had almost nobody on it but me. It's quite safe, and everybody's vaccinated now. But I just feel very lucky, and that some of the biggest changes in the trajectory of what I do have come when we thought an experiment was a total flop. Nothing happened the way we thought it should. And what did we do wrong? And we didn't do anything wrong. The animals are trying to tell us that we were on the wrong track. And so we would eventually get shifted and keep going. So,
Yeah.
so it's
Well,
been
thank
fun.
you very much, Margie, for spending some time with me this afternoon. Thank you.
Okay, thank you. This is fun.